Should I wait until Okay, should I say a few words first, just about what Sapiens is? So, hello? Maybe not yet, a little bit later. Hello. So if this is on, if it's not on, maybe the room is small enough and you'll hear my voice anyway. I've just been asked to give a few words to introduce our speaker today, Jovana Milic. So Jovana is one of my fellow PhD candidates at the SapienCE Center over at the AHKR, which is a center that focuses on early human behavior in South Africa in the Middle Stone Age. A center that's now getting to the halfway point of 10 years. It's an SFF center. And Jovana's project is, well, she'll be telling you about it today, but it centers around climate modeling based on shellfish or seashells and sea surface temperatures. It's a bit of a tongue twister. And yeah, I will leave it at that and give it to you. Oh, sorry. I don't know if you need this one. I'll just give it back to you.
So yeah, you heard already some introductory things from Ulla about Sapiens. I just want to say that that's like a multidisciplinary international project where we are concerned with research teams that are related to aspects can be actually broken down into more specific categories such as art, personal ornamentation, technological advancement, and the ability to expand into a wide range of environments throughout Africa and then ultimately globally, like in the world. So where does my research fit in here? Or what is my piece of the puzzle within Sapiens? So my project officially falls under the archaeology wing, but I like to think of it more as a hybrid between climate and archaeology because I am using archaeological materials, which are shells, archaeological shells, but to obtain proxy records from these shells to reconstruct paleoclimate. Paleoclimate. And I'm going to explain how that is done. But first, I want to have a look at what kind of information we can obtain from archaeological shell material about the past. So shells are a valuable resource for archaeological studies. First, maybe I can start with food habits because shells were mostly, they ended up mostly at the archaeological site as a product of food waste, product of human diet based on shellfish. So they were a common type of food, and we know this because we find them in Middle Stone Age context at archaeological sites along the coast of South Africa. And depending on the level of preservation, it's very useful to distinguish different shellfish species in the assembly because it can tell us something about the food preferences, the food and what types of shellfish were available in the past. And so sometimes it can show the preference, but sometimes it will show, it will show like dependency on what kind of resources were, or what kind of species were available based on the environmental and climatic conditions back then. So aside from representing the food discard, archaeological shells can be present in the form of artifacts, such as scrapers or beads, like you can see here, and we actually find these beads in the archaeological assemblages of Middle Stone Age sites, and they're actually one of the things that falls under this symbolic behavior package of early modern humans. But the role of shellfish in past coastal economies can also be evaluated through seasonal patterns of shellfish collection, and consequently we can deduce from that something about the site occupation. Seasonality is important for hunter-gatherers because it will affect when the food resources are available, and then acquiring of food had to be thought through, it had to be planned. If we have conditions that are not the same throughout the year, but we have they're very, like, we have different seasons, you will have to think and plan when that certain type of food will be available and how can I get it. So shellfish have been identified as a seasonally critical food source and a required source of carbohydrates and proteins during lean seasons, meaning when other food resources were scarce, people were turning to shellfish collection. But I will later show you that this isn't always the case, because it seems that humans sometimes harvested shells out of preferences, and just because they could, they were available, they were easily attainable, and not necessarily only out of pure need. And in the end, for this part, I also want to mention that we can use shells, archaeological shells, to date the archaeological sites. But of course, we cannot use it for the Middle Stone Age sites, because our sites are usually older than 50,000 years, and that is the limit for the radiocarbon dating method. So, for my research on shells, on archaeological shells, I'm relying on principles of sclerochronology. What is that? The term is derived from the word dendrochronology, but instead of studying the trees and tree rings, sclerochronologists study the hard parts of marine organisms like shellfish, corals, coralline algae, and parts of fish, otoliths specifically. It turns out that, like trees, these organisms produce growth increments. We use the term increment here, or growth band, instead of the ring, the tree ring. And they produce these throughout their lifespans. And within these increments, they continuously store the information about the environment they live in. Since I'm using shells, it is very important to understand how they are formed. And to understand how they are formed means, for me, to understand when I have a shell, where I need to sample it and where I'm looking, like how will I interpret the data that I get out of it. And before I started this PhD, I hadn't had the opportunity to work actually on shells, so learning about their biology when I started this PhD was I was amazed. And so I want to, maybe I can show you what was so amazing to me about their biology. But then I will show you like how they grow, how shellfish grow their shells. So what we call a shell is actually an exoskeleton of snails, clams, oysters, and many others. And their shells are composed mostly of calcium carbonate, and mostly like 98%, let's say. And that is the inorganic part. And then we have proteins, but that's like a very tiny fraction. It's like around 2%. So the body of a shellfish has this layer on its back that's called the mantle. And the mantle tissue, as you can see, lies just beneath the shell. And in contact with the shell, the mantle secretes, you can see, it secretes the proteins and it secretes then the minerals. And you can imagine it like laying down a steel, which here would be proteins, and then pouring over it a concrete, which would be minerals to fix it. So the shells cannot shed the shellfish cannot shed their exoskeletons, they cannot shed their shells. So they need to, since they cannot shed it and they need to grow, they need to expand it. And they do this by constantly adding new layers, and they add the layers on the margins of the shell. So that means that the newest part of the snail shell, for example, is located around the opening where the animal pokes out. And the outer edge of its mantle continuously adds new shell at this opening. And when you think about, why is that amazing? Because, for example, when you think about us, we grow our parts of our body in a different way. For example, think of the nails. The nails, they grow like from, you understand what I want to say. It would be like I wanted to add a layer on this part of my nail. So this is how they grow, in the opposite, from bottom up. So these new layers that are added on the shell, they will usually be larger when the shellfish is younger, because they tend to grow more faster when they're younger, and then the growth will slow down as they get older, and the layers will become narrower. But the width of increments can also vary in relation to other variables. For example, to temperature. Let's say some shells will grow broader increments when the temperatures are higher, for example. And apart from the width, we can look at the increments chemistry, and the chemical composition of shells can also vary with temperature or with the chemical composition of water in which they live in, among other things. So in sclerochronology, the shell increments are sampled and studied for various reasons, and here are some of the applications. First, I will mention ecosystem variability. What can shells tell us about that? So we all know that the present-day marine system is challenged by the combined impact of climate change and industrial-scale fisheries. And although these shifts in marine systems occurred before industrial fisheries due to natural climatic variations, they did not happen in this context of steep trends that we see today in ocean temperature, ocean carbon dioxide, and selective harvesting of key species. And to maintain ecosystem resilience under such conditions is a challenging task, and a key part of the process is to assess the degree of ecosystem variability that characterizes a resilient system. So what does that mean? It means, I'll try to translate it. How many species, what kind of diversity of life we need for us to say that this system, this ecosystem is able to thrive and survive. So this variability can be then assessed with the help of biochronology drawn from multiple sources, which I mentioned, like shellfish, corals, fish chocolates, and for shells, concretely what we observe are the indicators that we can observe in the shells are what kind of what impacts do we have on the growth rate of certain shellfish species? And this is a complex indicator because we have differences in food availability in shallower waters and food availability in deeper waters. Also, we have predator-prey relationships that can affect the growth rates. For me, the most interesting indicator from the shells that we can have that can tell us something about ecosystem variability is the environmental DNA. And why? Because when we take the DNA from the shell, we don't get just the host DNA or the shell DNA, because shells are able to incorporate microbial DNA also from the water, and also pathogen DNA. So we can get these things as well from the shell, so that's very useful. And we have stable isotopes as well, but I will tell more about stable isotopes later on. And so then when we have this data from the shells that some of the indicators that I mentioned, in combination with everything else that I also mentioned, such as other organisms, corals, corallinology, and fish otoliths, they can then help us to develop the detailed timelines of ecosystem variability and understand what happens at annual and seasonal level between different ecosystems and different parts of the same ecosystem. Another important application is in the form of environmental monitoring and reconstruction. Because reliable monitoring of past and present environmental conditions is essential when we want to accurately assess the impact of anthropogenic and natural changes on the marine environment. And shells can provide a tool for present and retrospective monitoring, establishing pre-impact environmental baselines. So I can give you an example. When we using the shells, we can, for example, track and observe the pollution events. We can, for example, compare shells from sites that we know are contaminated, let's say with metals, and that are not contaminated, because shells also tend to incorporate metals inside, so we can track that too. Further, we can apply sclerochronology in the service of climate. Data obtained from the shells can be used in climate modeling to create useful projections to inform ocean mitigation policies, especially now when we need it more than ever. And shells can then provide high-resolution data useful to project regional change in the short to medium term. And policymakers are usually most interested in these short to medium term deadlines. What can be done in short or medium term timescales. And one of the examples is that also an important issue that we are facing today are the elevated emissions of carbon dioxide in the atmosphere. And the ocean acts as a buffer against atmospheric carbon dioxide, taking up to 35% of it. And here again, we can turn to stable isotopes in the shells to measure which part of the ocean or spatial temporal variations in the ability of ocean to act as a sink for CO2. And now we finally come to the applications in archaeology that we are here today for. But I think it is great to show that sclerochronology is not just a way to collect information about the past, but also to tackle the important issues that we are facing today. So here I listed some of the geochemical proxies that can be used on archaeological shells, or that we can get from archaeological shells. What is a proxy in this context? Proxies are physical or chemical properties that we can measure from biogenic
so in my case shells, or sometimes abiogenic structures like sediments or rocks. And then these proxies can be interpreted as a signal from the environment at the time that the structure was formed. So we can call shells like a proxy archives. Out of the proxies that are listed here, I would say that the most commonly to date used one is stable oxygen isotope ratios. It is a proxy that I'm also using in my research. And I use it, it's commonly used to reconstruct sea surface temperatures, so the Paley temperatures of water. And then from that we can also infer about the season of occupation of archaeological site. So I would say the tricky part with stable oxygen isotopes is that it's not only dependent from the temperature of the water, but it's also dependent on the oxygen composition of the water. So if we want to reconstruct the temperature from stable oxygen isotopes of the shell, we need to know or to assume what the stable oxygen isotopic composition of the water was in the past. And that's not always straightforward and easy. Some of the things that need to be addressed when working with archaeological shells are diagenesis and pre-depositional treatment. Pre-depositional treatment is usually exposure to some sort of heat because shells were food, so they were usually cooked. So the genesis can be quite varied. It can depend on the type of the site that we have. It will be different for open air sites or sites that are caves and rock shelters. But it also it can be very dependent on the local factors that you have on the site and in the proximity of the site. And why are these challenging? Because both the genesis and heating of the shells before their deposition can result in their altered chemistry. So that means if we use these shells that have altered chemistry, when we use them for paleo environmental or paleo temperature reconstructions, we will not get the accurate results. We'll not get the accurate reconstructions. So that means that we have to find a way to somehow understand which ones have been altered. And sometimes that's easy because when you observe a shell, you will see visually that it has been burned or that something happened in the sediment, like it will look different than it usually does. But sometimes these alterations are not, we cannot see them. They're on a chemical level or mineralogical level, and then we have to find a way to check. And here we turn to mineralogy of the shells. And I told you that shells are mostly like 98% inorganic material, so mostly minerals. And shells are most commonly built of two minerals. They're called aragonite and calcite. And the one that is, I would say, more problematic is aragonite because it's more unstable. It's very easy for aragonite when it's heated to switch to secondary calcite. But then the good thing is if you have a shell that's originally aragonitic and you check it on one of these instruments that I put here on the slide, and you detect calcite in what you know is originally aragonitic shell, you can discard this one because you will know that it has been, that the mineralogy has been altered. But the relationship between mineralogy and chemistry is not always straightforward and it needs to be additionally checked. But yeah, I can talk a lot about it, but let's move forward. So now let me finally introduce the shellfish species I have been working on. This is the turban shell or Turbos hermeticus. It's a gastropod shellfish endemic to the south coast of South Africa. You can see that it has a shell and then this smaller round part that's called the perculum, and that's actually the part that we use for the analysis. Why? Because it's robust and it doesn't fragment as easily as the top shell. So consequently, because it's like smaller, it's more robust, it tends to be best preserved component of archaeological shell assemblages in the Middle Stone Age. And opercula are also more frequently found in the Middle Stone Age archaeological assemblages because sometimes I will mention this also later, but if the coast was too far, people chose not to take back to the cave the top shells because they're big and heavy. So just the opercula were transferred back. And that's why we have opercula in most of the Middle Stone Age layers, whereas we can't say the same for the top shell. And we are also lucky that turbo is able to build their shells and opercula in equilibrium with seawater. That means that what we get, the proxies that we get from this species will accurately reflect the conditions in which the animal lived. And that makes it well suited for paleotemperature reconstructions. I'm analyzing turbo opercula from the Blombus cave to reconstruct the temperatures of the water in which turbo grew and to determine the season of the occupation at Blombus cave. You can see here where it is. So how do we determine the season of collection? Here you can see a graph that shows plotted stable oxygen isotope ratios of 1 turbo operculum. And this is actually a nice looking graph where it's easy to spot the minimum and maximum values. We convert stable oxygen isotopes into temperatures when we put the values into an equation, but I will not talk about that because that's more complicated and they told me I can't talk about hard science here. So what I can explain easily is that the stable oxygen values are inversely proportional to reconstructed water temperatures. So that means that these maximum peaks that you see here with the red dots, they will translate to actually the coolest the coolest periods that this operculum recorded. So I mentioned earlier that the last added layers on the shell lay at its edge, and it's the same for the operculum. So you see the first few, let's say the first 3 samples on this graph, they will be the one to tell at what season was this particular shell captured. And you can see, because I told you it's inversely proportional, so this down, let me see if I can show. This is summer, and this is summer. So we see that this operculum, it's pretty straightforward when you have a nice graph like this. Like you can tell that this particular shell was captured during a warm season, so during summer. And yeah, this is just for me to try to simplify how we go from chemistry to archaeological interpretations. It is usually, as I said, straightforward with an identification of cool and warm maximums, but it's a little bit more complex when we have to differentiate the signals from the transitional periods, spring and autumn. So the annual timing of shellfish harvesting at Blombus may be indicative of foraging behaviors and social organization in a broader regional context. And such reconstructions based on the shells, and particularly on Turbo Cermaticus shells or opercula, already exist for the 2 sites that are close to Blombus. Pinnacle Point, that's around 100 kilometers away from Blombus, and Classius River main site, which is around 300 kilometers away from Blombus. Pinnacle Point actually contains one of the oldest shell middens or shell assemblages detected in South Africa and globally. It's dated around 164,000 years ago. And for Pinnacle Point, the shell data showed that the shells, the shellfish, were collected mostly during cool seasons, during winter. Whereas we have a different story for Classis River. We have a year-round season of collection. So why is that? One possible explanation for this could be related to the position of the two sites relative to the proximity of the coastline. Because there were, when the sea was lowered, Pinnacle Point could have been up to 10 kilometers away from the coast, whereas for Classis River, this is very different. It was much more closer to the sea, even when the sea was lower, due to the geography of Agulhas Bank. So we see, so I said for the Pinnacle Point site, we have a cool season collection signal, but we see that in the periods of interglacial-glacial transitions, we start to get a more year-round signal of collection, and that could also be indicative that as the environments or climate was changing, the subsistence strategies also had to change. So we still don't have the season of occupation based on shell material for Blombus Cave, as I have recently finished these analyses and I'm still waiting to get all the data. However, the seasonality reconstructions at Blombus Cave will certainly contribute to the interpretation of these ambiguities in the knowledge of the Middle Stone Age shellfish foraging strategies. And I chose turbo-opercula. I tried to choose opercula from strategically from those layers that can be compared to the other two sites. Also, although they don't match entirely, they don't overlap entirely because both Pinnacle Point and Classius have periods or layers that are older than Blombus. But here is what we know about the mode of occupation from different phases at Blombus so far. And these interpretations were mostly based on geoarchaeological and faunal data. So the long MSA sequence, Middle Stone Age sequence, was divided into major phases like M3 phase, lower M2 phase, upper M2 phase, and M1 phase. But then the M3 phase was further subdivided into three categories. The three phases that exhibit the highest densities of archaeological material are M3 CI phase, upper M2 phase, and M1 phase. Particularly important and interesting are upper M2 phase and M1 phase, because these are the phases where we have new techno-complex called Steel Bay, which is also one of that package that I told you that defines a behaviorally modern human. So, in contrast to these three phases that were high-intensity occupational phases, the other three have much lower intensity occupation. So let's have a look at these three phases during which the cave was extensively used. M3 CI phase seems to have been the period of the most intense human occupation. Human activities inside the cave during this period were stationary in their nature, such as they had on-site lithic production of tools and grinding of ochre on massive stones, and the sea was the closest that it ever was or will be was definitely to Blombos, because it was between 1 to 2 kilometers away. And this reflects in the abundance of shell remains that we find in this phase. It's the phase that has most abundant shellfish assemblages. So how they interpreted this phase based on geoarchaeology and faunal analysis, it seems that we have an intense and long occupation of the cave, but it was not so frequent, which is in contrast with the upper M2 phase, where you see that we have more frequent but shorter visits. However, shellfish densities drop significantly in this phase, and this could be due to the fact that sea was getting further away from the cave. So in this phase, it was almost up to 8 kilometers away. And we have a different type of use of cave space in this phase. We see that it was more organized, because we see that almost nothing happens in the central area, because we don't find the material in the central area, but more like on the peripheral sides of the cave. And the M1 phase, it's to some extent continuous to the upper M2 phase, because it's continuous in the way that the lithic technology that emerged in upper M2 phase is now fully established. But the shellfish densities are the lowest in the entire MSA sequence. And this also is probably related to the distance from the sea, which is now really drastic. It's the furthest that it has ever been in the sequence. It's almost up to 44 kilometers away. So this is where we actually don't find the top shells of turbo in the layer that I mentioned before, because the sea was so far away, so people probably chose to discard the shells at the site of the collection at the coast and take just opercula and flesh back, because opercula and flesh is connected and it's not easily detached when it's not cooked. So about this phase, there's no doubt that this was a high intensity occupational phase, but we have conflicting interpretations at the nature of this intensification, because some argue that the cave was inhabited for longer periods, whereas others argue that this intensification is actually due to increased mobility, residential mobility, and frequency of the visits. So all Blombos Cave Middle Stone Age phases of higher occupational intensity correspond well to the shifts in vegetation, sea levels, and subsistence, indicating that hunter-gatherers had to adjust their mobility strategies. So to determine the season of collection or of site occupation will contribute to a better understanding of the variations in settlement patterns throughout the sequence. And the seasonality investigations will shed more light on conflicting interpretations that I mentioned about the length of occupation for the youngest two phases, in which important cultural and technological innovations happen. And I think I will just mention that in my opinion, one of the biggest advantages of using climate records from the shells compared to data that we get from the marine sediment cores is the possibility to have high-resolution sea surface temperatures for coastal locations. So the data that we obtain from the shells, they are very, very local. And we can then get a better idea what those early modern humans really experienced, like when they entered the sea. And it's just that the shells, archaeological shells, they are directly related to archaeological contexts. So I think this is their greatest advantage, and it will be, I can't wait actually to have all the data and to see what's happening with the season of shellfish collection at Blombos in the sequence, and to compare it with the data that exists for the region. So thank you very much for listening. I hope it was not too sciencey and that you can understand, but I'm here to, if somebody has questions, I'm here to answer if I know the answer. Thank you. Yes, do we need to give you a microphone?
Get a microphone from me.
Oh yeah, okay. I can speak loudly anyway. Thank you very much. Very interesting talk. I have a question about the amplitude of the temperature records that you use clumped isotopes, as far as I understand, for temperature.
I don't use it, but you can use clumped isotopes. Yeah.
I know that's one of the problem, that is quite big uncertainties in the temperature estimates by the clumps as isotopes. But so the question is, what is the present day seasonal temperatures variations? And you see changes, because these are early ice age environment. Is it the same order of magnitude for the seasonal changes?
I would say it's very similar.
Yeah, and what many degrees are we talking?
I think around 5 degrees.
5 degrees.
Yeah.
Okay, so okay.
Yeah, and based on what I've read from the other sites, I think it's not that different.
So you see same amplitude.
Similar, yeah.
And another thing is a kind of detailed question though, but you said that you estimated the distance to the shore, to the collected. And I presume, because I saw that you correlated this with isotope curves, which again sea level, global sea level curve. So what actually you did you estimate the sea level based on sea level curves and found the distance in that way. Is that the correct way? Because you're saying that it was a long time during isotope stage 4 or a transition to 4, whereas other periods it was shorter distance.
Well, I can tell you what we do in Sapiens, because we are also trying to do this with the proxies that we have from our research. So we get different kinds of proxies that we put into models, into climate models, and then that's one of the ways you can estimate what the distance was of the sea to the site. Not sure if I answered your question.
I can perhaps answer.
But yeah, this is my supervisor, and she.
Do not put the data into climate models.
I meant sea level.
Yeah, sea level reconstructions and the distance from the site today is made by other people. Helicopter, for example.
Yeah, right.
Yeah, so
but that is the methods that is used anyway. It's correlation with sea level data.
And marine geological investigation.
Okay, I see. Thank you.
This is not something we have done in Sapiens.
No.
So good to have her also here. She's a true sclerochronologist. She's not an archaeologist who is trying to do sclerochronology.
Can I ask a little more question?
Yeah.
How old are these oldest shells? I mean, how many years can they be?
So from Blombos, the oldest are around 90,000.
Yeah, but I mean the living. How old?
Oh, how old my shellfish species can live? Up to 10 years.
Oh, so it's only 10 years.
Yeah, it's only 10 years.
Okay.
They're not long lived.
Okay.
Can I follow up on that one?
Yeah.
So when we're talking of this biochronology, is it also possible to, within the species, to make like wiggle matching or find actual layers? I mean, it's an enormous time span when you excavate these type of caves. So even 30 years is like it's just some seconds in
it's
some
nothing.
sense. Yeah. But
is it possible to find and thus also prove the ability to reconstruct environments on that on like an annual basis, so to speak? Do you understand? So if you have several samples and you actually find the same, like we do in dendrochronology.
No, you can do it in other shellfish species, but they have to be a lot more long-lived.
Oh,
So
okay.
10 years is certainly a lot too short. I mean, it's like Arctic Icelandic is the species of course here, and that can become several hundred years old.
Because it would be interesting from an archaeological perspective, of course, to see if you actually collected species from the same.
Actually, from South Africa, from our side, we don't have any long-lived shellfish species,
Okay.
so they're all mostly short-lived.
Things that are this short-lived. But you can certainly get sort of, if, for example, this would have been in a younger time period where you can use seafood in dating, you can then have short floating chronologies.
But you mentioned corals. You at the introduction you mentioned you have algae that goes
Red
causing
coral
crusts.
algae, yeah.
Do you find that there? Because I presume they may give a longer record.
No, we don't have.
You don't
To
have.
my knowledge, we don't have it
Okay.
in Blombos. Yeah.
Okay.
So that coral algae, there are no point for people to collect coral algae. Well, there are no crust-forming coral algae.
Toriel?
Det er forskning, ikke for...
forskningen. I was wondering, how far did what is the furthest, the longest distance people carried these shells? Because that would tell us something about what people really like to eat if they really carried them long.
So, I mentioned, at least for Blombos, the coast in the latest MSA phase was up to, more than 40 kilometers away. And we still find shells back at the cave. So that's a long way to go and come back and bring the shells back. Yeah,
But were they eaten, or were they playthings, or were they carried there by animals or birds?
No, Turbo definitely needed to be collected and brought back to the cave. They were definitely eaten. It's just like, as I explained, when you have to walk such a big distance, it really doesn't pay off. It requires so much manpower and strength to carry these big shells back that are really heavy. So this is why they still definitely like to eat them, or they opted for it, but they had to strategize how they're going to bring them back. But they were definitely eating them. It's primarily the food source.
That tells us something about preferences, and perhaps these were sacred food in some way. They had a special meaning for them.
But maybe they didn't have anything else to eat. Maybe they didn't have anything else to eat.
Maybe they didn't have anything else to eat. This is what Karen is saying.
Oh yes, they did.
So they had to do it.
I'm sure they had.
Perhaps a question on the side, but since you mentioned that, your supervisor mentioned that radiocarbon methods, have you investigated the limitations of the radiocarbon methods related and in relation to thorium on shells in the time depths you've been working with the Blombos?
So we also have later Stone Age deposits that are much later and than 50,000 years. So this can be then investigated to see if we can use radiocarbon method on this. We cannot use it on MSA because the error would be enormous because it's so much older than 50,000 years. And the uranium thorium, we haven't done it on shells. We've done it with ostrich eggshell. And that that showed to be a promising method, but we haven't done it on shellfish, on shells.
The short answer is no, and it's probably not an option because in most shellfish, uranium thorium is an open system. It won't work. It doesn't work like in cars. You can't use uranium thorium to date bivalves or stuff.
So dating methods on the shells from this time
The dates on the shells are entirely based on the you don't date on the shell.
No, okay. So you never.
Potentially amino acids could be used for dating because you know if you know the temperature history, you can. The other variable is the age then.
One thing that we could do, and what I'm thinking about doing with this, now I'm taking over, is that because these little orphoculums are small in time, if you have them evenly distributed over a long time period, as Classes River, for example, we have -5, then you can use the sea level curve and these little short windows in time and try to wiggle match to improve the age model for the cave.
Yeah, but this needs to be tested. Maybe it will not work.
I just had a short comment to the lady, but I think she went. But my experiences from, for example, Peru is that you can carry shellfish a long way because if you keep it alive in a net, it's a perfect source for also water. So in the Atacama Desert, you find shellfish a long way in, even longer than perhaps like 100 kilometers. So I think they.
So how big are your shells?
Oh, I mean, I can't remember now, but I mean, there were really big shells as well,
Okay,
but
yeah.
this is clams more.
Oh,
So
okay.
it's not the same type. I just wanted to return to what you started to talk about, like with the shore levels, and could we use your method instead to actually get better shore level, a better system on the shore levels in that area? Because I've been working a little bit with coastal shelf archaeology, and it's quite hard in these areas actually I mean, we can have the global models and so on. Perhaps you have better data now from marine geology, but just a thought that struck me, if you have all these caves and you have pretty good datings and the biotopes of the gathered shellfish, it would be really interesting to see if we could get some more fluctuations, because these are like these coral curves and they are yeah. So this could perhaps be a proxy for shore levels as well.
Yeah, I think
like the
then we're perhaps talking more about different species being present in a
okay.
site, but we can talk after.
Yeah.
Thanks. This is a naive question, but like what was so cool about that cave?
Sorry?
What was so cool about that cave? Like, I don't know, like it seems to me like if I'm walking 40 kilometers with the shellfish, because I want to go back to that.
You mean what was so cool about it in the past?
Yeah.
not today.
Yeah, no, but it's just like, I feel like if I'm walking 40 kilometers towards I'm going to encounter something that'll give me shelter along the way, aren't I? Like, is it like really there's no other place there? Like, why were they going back to that one? Like, because there's a bunch of stuff you guys found over the years, right? So like,
Yeah.
is there like a bunch of other ones and you guys are just like, no, we like Blumbos, that one's cool, it's close to the airport? Or like, is that like the only, like, why did they go back? So sorry, yeah, it's a naive question. You guys did your scientific ones, but like, I'm coming from another department type thing, and I don't get why I'm hearing about Blumbos all the time. So why Blumbos is my question.
Oh, it's, I'm not sure, but I've showed you some of the closest caves from that period, or like partially overlapping period. So I don't know. For some, like this gentleman here, 100 kilometers is not that far away, but maybe for some it was. So then, if you have a next site, the next closest site that is cave. Refugium that is like 100 kilometers away or more, Ole can say something about this.
I can maybe shoot in a little bit. Just I don't know if I need that. I can hold it. It's the talking stick.
It's for the screen.
That was for the screen. Okay, apologies then. No, but you asked, like, back in the past when people, why did they choose to go all the way out 100 kilometers from the coastline? And when you looked at the map that Jovana presented, there are just a few spots that we have these Middle Stone Age sites that are investigated. And there is a thing where all along the coastline here, we have a lot of archaeological material, and quite a lot of it is also, you know, like artifacts that could be Middle Stone Age, but they are in open air sites. And so when you go in as an archaeologist to try and investigate, you want a context that is fairly secure. And in fact, there are not so many of those. So we kind of pick the little peak holes that we can get that are secure, and we investigate those. Potentially on this Agulhas Bank, there are a bunch, I mean, like endless amounts of sites that are under sea level, but they are not very easy to go and look at. So it's this.
Yeah, but maybe what he meant is like, if you do have sunk sites, like why did they still choose to go
all the way?
All the way back to
That's a good
Blombos?
question, right? Because it is this thing like, are we looking at two addresses in Bergen and interpreting the whole Middle Stone Age based out of that? Potentially, right? We are, because we don't really have that much of a choice. So yeah, are cave sites outliers or are they the norm? And quite possibly they are outliers. That's very hard for us to say. So it is a good question in that regard, and it's one that's a bit hard to answer because we only have limited keyholes to look into in a way. Yeah. So I don't know if I can give you a definitive answer, but it's a hard period to look into like that.
And it's combined with what she was saying, that maybe they had no other food source or something.
It could be that, right? You know, this. What is the role of a cave site in an environment like this? It's definitely there are.
No, actually, in that period that I was when the coast was the furthest away, they had a really broad subsistence. So that's actually not an explanation. It was not. The shellfish was not the only food they had. So that's not an explanation.
Have you guys eaten? Just tried it?
Yes,
We did.
It's a dish in South Africa.
Okay.
And then people go out foraging by themselves and they cook. It's a common thing.
Okay.
So common that you have to limit the number of specimens people forage each time. You have to have a shellfish permit.
Ah,
Okay?
cool.
You just have to mince them up because they're a bit tough.
Yes, Aliki.
Yes. When you showed us the different strata, I don't know how to explain. Yes, here. You mentioned that in M3 you had a lot more shellfish than in M1.
Yeah.
Does that include the ornamental shellfish as well? So people way, way back in the past used shellfish more often as ornaments than in M1 period, for instance.
This I'm not sure about.
So in M1, have you found anything that could be considered ornamental?
In M1?
Yeah.
Yeah.
So they still might have brought back the entire shell with them just to use as an ornament.
But the shells that were, at least those that were used in Blombos, they're really small. So it was not, they're tiny, so it was not a big problem to bring those back.
Okay.
But we don't have, as of now, any evidence that these big shells that were primarily food, that they were also used as some kind of ornament.
Can I just
Karen?
add one thing? The shells that are used for the bead necklaces and stuff, those are not necessarily marine.
Yeah.
So they live in shallow rivers, so it can be brackish water.
Oh, I see. So, and the river was...
Yeah, I mean, on the coastal plain, there are several
Yeah.
rivers and waters.
Okay, and So would you happen to find more that came from the rivers than those that came from the sea? Or is such a comparison not really possible?
I mean, I'm not an archaeologist, so I don't know which layers the shell
What did
bits are more
you
abundant.
ask about, Ricky? Sorry, I couldn't hear you.
So I was wondering if you happen to make a comparison, or if such a comparison even is possible, that more shellfish that were used for...
Oh, it's those that were food are more abundant, if that's the question.
No, like if it's more common for shellfish to be used as ornaments if it comes from the river versus than if it comes from the sea. So if it comes from a location perhaps closer to the cave than if it comes from a farther away location.
I'm not sure about that.
I guess that the Nasaris that they use for the bead necklaces, you can find, they could have found them a lot closer to the cave.
Okay.
Compared to the seas, it doesn't have to be open ocean marine conditions.
Yeah.
I know there is a species of shellfish from Middle Stone Age layers called Donexera. It's a bivalve, and they not so extensive. Like it was not a habit, but some of them had perforations, and then they were. But this is not like something you encounter commonly. But then they were like the people who are doing studies on this. They are theoretizing like were they really an ornaments, or maybe something that was more for practical use, like putting it. I think they were mentioning something around your feet or like legs, and then it would make a sound as you go. It would something like that.
That's very interesting, though.
Yeah, but I'm not an expert. It was just a lecture that I attended, and I remember this because it was so interesting, and because before that I haven't heard, at least I don't think from Blombus we have perforated Donexera. Yeah, Larissa.
Maybe I can just add, because your first question was whether in M3 they are found, but there are none found there. So the earliest beads, they come from around 70, so from M2.
Yeah.
As far as I know, or at least the published ones.
Also the ones that were not.
Yeah, and they are not, so that's particular about them, that they are tiny and they are not abundant, and they were not used for food. So they were taken to the cave particularly for another purpose, we think ornamental, but yeah.
Can I just ask? Yeah, thank you. These shell ornaments, are they something that appears at the same time as the Still Bay complex? Yes. So it's considered a sign of behaviorally modern humans? At least that's how we theorize it. And just one more thing that I was wondering a little bit about during this talk. What is actually the nutritional value of these shells? I mean, are they sort of high in nutrition, or how does that work?
Well, not that high, but you can get let's say enough carbohydrates and proteins from them, but I don't think they would be enough for you to go on them, just based on, to have a substance just based on that.
So if you walk, say, 40 kilometers to the coast to gather them, you actually spend more energy than you get from the shell, essentially?
Yeah, but one thing is the energy, another thing is the nutrient. But in this case, you have, like today, we eat shellfish, we know we can get a lot of omega-3, for example.
Yeah, that makes sense.
So that's actually lots of studies dealing with different food times, and in this case, the importance for the development of the brain.
Does that answer your question?
Yeah.
Well, I'm so, as I told you in the beginning, Sapiens is like a mosaic, and you see we have people from archaeology, from climate, from psychology, and I don't think there's one person who can answer all the questions from these three fields, so then it's really good to have my crew here to jump in. Yeah, are there any more questions? Okay.
Well, thank you for coming then. And thank you for
Thank you very much for the attention and for making an effort to come and attend.